An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day, our bodies must produce millions of skin cells to replace those lost through normal activity. Each of those cells must have a complete complement of the genetic material. Prior to cell division, whereas the cell divides into two identical cells, the DNA needs to be replicated so that its daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This bit on a string structure is called chromatin. As a cell prepares to divide, chromatin calls up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replicated DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cells goes through the interphase, which can be divided into three distinct phases. During G1 or gap 1 phase, all the organelles and cytoplasmic components, including the centroids in the animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap 2 phase, all the enzymes needed to aid in the process of cell divisions are produced. Most eukaryotic cells spend a great deal of time in interphase and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton dissembles as the spindle begins to form. In animal cells, centroids play an important role in the distribution of the chromosomes in the dividing cell. The centroids migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attached by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes towards the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell called the metaphase plate. Anaphase begins by the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus dissembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cell. Cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells.